Okay, so far we have discussed about a few biochemistry experiments, uh, actually qualitative test for carbohydrates. If you can remember, we discussed about MOS test, that is for identify reducing sugars, and MOLIS test, that is to identify most of the carbohydrates, and then failing test, again, that is to identify and distinguish between reducing sugars and non-reducing sugars. And then we discuss about, I think, um, ozone test. And using ozone test, that is very specific and important test. We can identify the type of reducing sugar. Okay, so that's what we discussed so far. Today, I'm going to discuss with you about Barfoed test. Okay. Okay, the Barfoed's reagent is not reduced appreciably totally by reducing disaccharides uh, such as lactose and maltose, but it is readily reduced by monosaccharides. Therefore, the test is used to distinguish reducing disaccharides from monosaccharides. The Barfoed's reagent contains acetic acid. As a result, if the contents are boiled for a long period of time, there is a possibility of hydrolyzing the disaccharide into monosaccharides and thereby show reduction. Hence, prolonged heating should be avoided. Okay, so I just read the instruction, the introduction about Barfoed test. As you can see here, we can use Barfoed test to distinguish between reducing monosaccharides and reducing disaccharides. Okay, so there's a specialty of Barfoed test. Okay, so if we summarize what we have discussed so far, most test, it is a very preliminary test. I told you that nowadays we don't use that test. It is to distinguish between reducing sugars and non-reducing sugars, and there's a limitation of that test because if we do the prolonged heating, again, non-reducing sugars also can give positive results for most tests. And then MOLIS test, it's a test to identify reducing sugars, and you know that monosaccharides, reducing disaccharides, as well as polysaccharides give positive results for that MOLIS test. Okay, and not only reducing sugars but also non-reducing sugars. Okay, so both reducing and non-reducing sugars give positive results for MOLIS test. And then we discussed about failing test, that is very common test to identify reducing sugars. Okay, and then we discussed about ozone test, that is to identify the type of reducing sugars. Okay, ozone test, that can be used to distinguish reducing sugars and non-reducing sugars also, because non-reducing sugars will not give positive results for ozone test. So in that sense, we can differentiate reducing and non-reducing sugars. And the specialty of ozone test is that we can identify the type of reducing sugar using the ozone test. So this Barfoed test, there's again a specialty, because using this Barfoed test, we can identify the type of we can identify and we can distinguish between reducing monosaccharides and reducing disaccharides. Okay, this Barfoed's reagent will readily totally reduced by reducing sugars. You know that reducing sugars or reducing agents are any substances which loses electrons to other substances in a redox reaction and gets oxidized to higher state. Okay, that's the definition for reducing agent. Okay, so reducing sugars are the sugars that act as reducing agents. So we discussed about that. So in that sense, Barfoed test is used to distinguish between reducing monosaccharide and disaccharides. Okay, so I'll quickly go through this test. First, you have to take about 3 milliliters of the Barfoed reagent into test tubes. In here also we are going to use glucose, fructose, maltose, sucrose, and lactose. Okay, and we have to add that sugar solutions into that boiling tubes, and we have to boil that, and that after that we have to while boiling you have to observe the color changes. Okay, so if there is reducing monosaccharides, okay, present in the samples on the sample solutions, that will give positive results within about 30 minutes. Okay, so if the sugars we testing are reducing disaccharides, so that will take a longer time to give positive results. And also non-reducing sugars will not give positive results. Okay, so that are the intended outcomes of this research or of this test, or the intended observations of this test. So I'll show you the video practical recording. So after that we will discuss.
Your next practical is Barfoed test. It is one of the qualitative tests which is used for carbohydrates. For that test, we are using Barfoed reagent and also 5 types of sugar solutions, namely glucose, fructose, lactose, maltose and sucrose. For that test, we have to firstly add 3 ml of Barfoed solution into a boiling tube. I am using a measuring cylinder to measure Barfoed solution. I am adding 3 ml of Barfoed solution into each and every boiling tube. After adding Barfoed reagent, you have to boil the Barfoed reagent into few seconds. I am using a Bunsen burner and I am using the direct heating method. After heating, we have to add few drops of 1% sugar solution into this Barfoed reagent while heating, and also make sure we have to heat it only up to 30 seconds, that means half an minute. After that, we have to remove the sugar solution from the heat source and we have to allow it to cool. After that, we have to re-examine it. Now I am doing the test for glucose solution. First of all, I am heating the Barfoed reagent in few seconds. After that, I am adding sugar solution into the Barfoed reagent while heating. In here you can see after 30 seconds we have to remove the solution from the heat source. In here within 30 seconds you can observe there is a brick color precipitation in the boiling tube. Now I am proceed the same procedure for fructose. Before adding sugar solution, I am going to heat the Barfoed reagent. After that, I am adding the fructose solution while heating. Within 30 seconds, I mean before 30 seconds, in here you can see a brick color precipitation for the fructose solution. Then let's proceed it into lactose solution. Before adding lactose solution, I am going to heat the Barfoed reagent. After that, I am adding lactose solution drop by drop while heating. Now the time is up. In here, within 30 seconds, it cannot observe any sign of brick red color precipitation for lactose solution. Let's keep it in a test tube bag and let's examine it after few minutes. And this is the maltose for maltose solution. For this also I am adding 1% of sugar solution drop by drop while heating for 30 seconds. Within 30 seconds, for that also you cannot observe any sign of brick red color precipitation. Let's keep it for few minutes and examine it again. And this is for sucrose. For sucrose also I am doing the same procedure. Now the time is up. In here you cannot observe any sign of brick red color precipitation for sucrose solution. These are the overall observations for Barfoed test. You all know this is glucose. In here you can clearly observe a brick red color precipitation. It forms during the heating, that means within 30 seconds. This is fructose. It also forms a brick red color precipitation within 30 minutes. 30 seconds. This is maltose. In here, now you can observe a brick red color precipitation for maltose. But it doesn't give any brick red color precipitation within 30 seconds. But within the cooling, it forms a brick red color precipitation. In the case of lactose, it also doesn't give any brick red color precipitation during the heating. But during the cooling process, it also forms a brick red color precipitation. This is sucrose solution. It doesn't form any kind of precipitation during the heating or during the cooling process. By using this test, we can differentiate reducing sugars from non-reducing sugars. Also, we can differentiate reducing monosaccharides from reducing disaccharides.
So have you learned about Barfoed test before? Have you heard about Barfoed test? I think that's not in the advanced level syllabus. Okay, so test is much similar to the Fehling test, and this is the redox reaction in the Fehling test. You know that we discussed that in the last week. In Fehling's solution, it contains Fehling's A and Fehling's B. Fehling's A contain copper sulfate, and Fehling's B contain sodium potassium tartrate along with sodium hydroxide. And there's a when preparing the Fehling solution, we mix Fehling's A and B, and when mixing, it will form this tartrate to cuprate 2 as the active agent in Fehling solution. So in the redox reaction, Cu2 plus is acting as the mild oxidizing agent, and it will reduce to Cu plus, and the reducing sugars will be further oxidized into carboxylic forms. Okay, so and finally, upon heating, this cuprous hydroxide will form the brick precipitate, which is cuprous oxide. Okay, so that's the reaction happening in the Fehling's test. So similarly, and this is the redox reaction in Barfoed test. Okay, you can see in Barfoed reagent contain copper to acetate in 1% acetic acid. Copper to acetate in 1% acetic acid. Okay, even though the redox reaction is quite similar to that of Fehling's test, but you should know that the conditions we use are different. You know that Fehling solution is alkaline, has alkaline medium, but in Barfoed's region, it contains slightly acidic medium. Okay, so even though the principle is same, the conditions is different. That is why these two tests are considered as two different tests. And also our intentions or objectives of using this test are different. Okay, so basically Fehling test is to distinguish between reducing sugars and non-reducing sugars, and for Barfoed test it is used to distinguish between reducing monosaccharides and reducing disaccharides. Okay, you know that reducing monosaccharides have a higher reducing power than reducing disaccharides. Okay, so that is why reducing monosaccharides will give this brick color precipitation within 30 seconds or within a short period of time. But reducing disaccharides, they can give positive results, but it will take a longer time because they have lower reducing power than reducing monosaccharides. Okay. Yes, only sucrose cannot give precipitation after heating or cooling. Someone has directed the question. Yes, why is that? Even though sucrose is a disaccharide, it is not a reducing sugar. Okay. In order to happen this redox reaction, there should be a reducing agent as well as oxidizing agent. Even though we add the mild oxidizing agent as the Barfoed test to all solutions, there should be a reducing agent to occur this redox reaction. Okay, redox reaction when the oxidizing agent reducing agent දෙකක් තියෙනවා. ඒ කියන්නේ electrons දෙන්න පුළුවන් කෙනෙකු electrons ගන්න පුළුවන් කෙනෙකු. Okay, so sucrose will not give results for this test. Okay, so there can be, yes, another question. Why adding sugar solution while heating? So that's the differences. These tests are designed in that manner. Okay, and you know that when we doing any experiment or any research, we are doing a trial and errors, and we come up with the best. We optimize the test conditions, and we come up with the best conditions. Okay, so these tests are designed in such manner. So I cannot tell that why adding sugar while heating, because this test of the positive results, the intended outcomes of this test will occur if we perform according to the procedure. Okay, according to the standard procedure. Okay, so we cannot just alter the steps, and finally, if we do so, we cannot get the intended outcomes, or we cannot achieve the objectives. Okay, so in this test, that's why I told you that the redox reaction is so similar, but the test conditions are different and the reagents are different. Okay, in Barfoed reagent, it contains copper 2 acetate in acetic 1% acetic acid. Okay, so that is acidic medium, but in the failing test, it contains CO2 plus in basic medium. Okay, but the redox reaction is similar. Okay, so that's the that is what happening in the Barfoed test. And also you should know, primarily this test is to distinguish between reducing monosaccharides and reducing disaccharides. Okay, yes, that is a good question. Lianage posted that why 30-second heating. If heat more time, what happens? Yes, if we heat more time, reducing disaccharides also will give positive results within a short period of time. You know that Barfoed's reagent contain not concentrated acid but acids, acetic acid. Okay, if we do heat for a longer period of time, reducing disaccharides also will hydrolyze into monosaccharides. You know that disaccharides are consisted of 2 monomers or 2 monosaccharides. Okay, then it will hydrolyze into monosaccharides and will give positive results within a short period of time. So our intention or our objective is to distinguish between reducing monosaccharides and reducing disaccharides using Barfoed test. So if we do heat for a longer period of time, more than 30 seconds, so our intention cannot be achieved. So that's the answer for that. I think DNAG, I answered to your question. Okay, any questions? Yes, can you repeat what solutions direct will give precipitation? Okay, that precipitation is the briquette, is briquette color precipitation, that is the cuprous oxide Cu2O, and reducing sugars will give precipitations in this test. Okay, but the time taken to give positive results, it means the brick color precipitation is differ. Okay, reducing monosaccharides have a higher reducing power than reducing disaccharides. Therefore, Reducing monosaccharides will give positive results within a short period of time. Specifically, within 30 seconds, they will give positive results. It means the brick color precipitation. Reducing disaccharides, they will give positive results. That means the brick color precipitation, but it will take a longer time. Okay. Altogether, reducing sugars will give positive results. It means the brick color precipitation for this test. Okay, but non-reducing sugars will not give positive results. Okay, that's why sucrose haven't given any brick color precipitation in this test. Okay, because you know that sucrose is a non-reducing sugar. Okay.
Yes, sir.
Yes. Did you get what I said? What is the objective of Barfoed test? Can you say that?
To identify reducing monosaccharides and reducing disaccharides.
Yes, good, good. Yes.
Also,
Yes.
reducing sugars and non-reducing sugars.
Yes, good, very good. As she correctly answered, we can use Barfoed test to distinguish reducing sugars and unreducing sugars, and primarily to distinguish reducing monosaccharides from reducing disaccharides. Okay, so that's it. Then I will move on to the next test that we are supposed to discuss with you. That I supposed to discuss with you is Trommer's test. Okay. So in Trommer's test, that is very primary test. That is actually this test is firstly developed, then failing and Benedict test. Okay, and after that, failings and Benedict's test have been developed. Okay, මේ Trommer's test එක තමයි මුලින්ම ඇත්තටම මේ reducing sugars අඳුර ගන්න පාවිච්චි කරපු test එක. ඊට පස්සේ තමයි මේ specific reagents පාවිච්චි කරන්න ගත්තේ. Okay, so we have included this test also, but after learning this also, you will understand that if you are asked to use a biochemical experiment to distinguish reducing sugars and non-reducing sugars, you should use a better specific test. Okay, and even when we discussing about most tests, I told you that it is not to a specific test, and so similarly Trommer's test also it's not a very specific test. Okay, and this is very basic test. In here we have to take 2 milliliters of sugar solutions into boiling tubes. We have to add equal amounts of 40% sodium hydroxide, that means somewhat concentrated sodium hydroxide alkaline, and add a very dilute copper sulfate solution drop by drop and observe the color change. And we have to continue that in copper sulfate solution until a paint ability occurs, and we have to heat the contents in the boiling tube and observe the color change. Okay, so that's what happening. That's what we have to do in Trommer's test. Okay, actually in the past this test is used to identify the presence of glucose in urine. Okay, in the very past in the history, this Trommer's test has used to identify the glucose in urine. Okay, so you know that glucose should never be present in urine if healthy in a healthy person. Okay, if glucose is present in the urine, so it means there can be some adverse health condition. Okay, so in the past this test is a very simple one and has been used to identify the presence of glucose in urine. Okay, and now the more advanced biochemical tests have been developed, and I think that nowadays we don't use this test. Okay, so that is also basically to distinguish reducing sugars and non-reducing sugars. Okay, so I'll show you the practical procedure and we will discuss.
Your next practical is Trommer test. It is also one of the qualitative tests which is used for carbohydrates, which can be used to differentiate reducing and non-reducing sugars. For that test, we are using 40% of sodium hydroxide and very diluted copper sulfate, and also we are using 5 types of sugar solutions. First of all, I am adding 2 ml of sugar solution into a boiling tube. For that I am using a dropper. Then we have to add an equal amount of 40% sodium hydroxide. That means 2 ml of 40% sodium hydroxide for each and every tube. After adding sodium hydroxide, we are going to add very diluted copper sulfate drop by drop. And let's see what happened to the solution. While adding copper sulfate, you can observe the color of the solution is turned into somewhat blue color, and also in here you can see a unclear turbidity is formed. And proceed the same procedure for each and every sugar. I am adding very diluted copper sulfate until the unclear turbidity occurs. Here, after adding copper sulfate for fructose solution, the color has changed into somewhat greenish color. Okay. Others are remain at blue color. The color of the fructose is changed into somewhat greenish color. After that, I am going to keep these solutions in a water bath. While heating, we have to observe the color change. In here, for fructose, the color is changed into orange color. This color change is same as the Fairing's test. It changes into blue color into mud green color, after orange color, and finally it gives brick red color precipitation. In here you can see fructose sample is already changed its color into brick red color. The color of the glucose solution also changed. But the color of the sucrose solution does not change. It remains as blue color. By using the Trommer test, we can differentiate reducing sugars from non-reducing sugars.
So basically that test also to distinguish between reducing sugars and non-reducing sugars. So you may think that why I am telling about Trommer's test after discussing about that failing test and why this Trommer's test. Okay. Actually, the Trommer's test is, as I mentioned earlier, it is the firstly developed test to distinguish reducing sugars and non-reducing sugars. And Trommer's test, there's one specialty is that we don't use a specific reagent here. Okay, we just use sodium hydroxide and copper sulfate. Okay, sometimes in our laboratories that we may not provide it with failing solutions. Okay, you know that failing solutions are reagents. Okay, in failing say we have to prepare the failing solution. We should provide it with should provide it with failings A and failings B. Okay, so do say similarly in Benedict's test. Okay, in Barfoed test also Barfoed reagent should be there. Okay, unlikely in those tests, if we don't have any kind of that reagents, then if you need to identify or distinguish between reducing sugars and non-reducing sugars, so you can use this test. Okay, then if you have failing solution, Barfoed solution or Benedict solution, so you can use those tests. Okay, depending on the situation, you can use the theory. Okay, actually this Trommer's test, as I mentioned earlier, is heavily used in the past to identify the presence of glucose in urine. So if, as I mentioned, glucose is not present in urine in normal conditions. So if if an individual suffering from diabetes or something, so glucose can be there. So it's a sign of diabetes. So it has been used in the earlier to identify or to test diabetes. Okay, now we have, we don't use that test. We can simply using a glucometer, we can measure qualitatively the blood glucose levels. Okay, so that's the history and the theory behind Trommer's test, and the reactions, the redox reactions in Trommer's test is similar to that of failings test. Okay, and here we use we add sodium hydroxide and copper sulfate basically. Okay, then it will form cupric hydroxide, and upon heating, cupric hydroxide will form this cupric hydroxide will reduce to cuprous hydroxide when there is a reducing agent. Okay, so you know that reducing sugars has a reducing power, and for an example, if glucose is there, it will oxidize into gluconic acid, and CO2 plus will reduce into Cu plus, and this cuprous hydroxide upon heating will form cuprous oxide, that is the brick red color precipitation. Okay, so as you can observe those three tests, failings test, Barfoed test, and the Trommer's test. Those three tests have finally the same redox reactions, but the conditions we use is different. Okay, reagents, the acidity levels, and the conditions we supply is different. So in Barfoed test, we do specific, we do heating for 30 seconds. In Trommer's test, there's no specified time for that. So such like that differs. The conditions and the reagents differs, and the objectives are objectives also differs. Okay, did you get that? තවබරි හිතන්න පුළුවන් මේ හැම එකේම එකම දේ වෙන්නේ මම මගුලත් හැම එකේම එකම දේ වෙන්නේ මොකටද මෙච්චර ටෙස්ට් කියලා. ඒක හරියට තේරුම් ගන්න. දැන් හිතන්න ඔයගලන්ට ලැබ් එකේ failing solution නැත්නම් Barfoed solution එක නැත්නම් Benedict solution එක නැත්නම් reagent එක නැත්නම් අපිට reducing sugars අඳුර ගන්නම ඕනේ නම් මේ වගේ simple test එකක් පාවිච්චි කරන්න පුළුවන්. Okay. Actually, when we using Trommer's test as a clinical test, we will observe series of color changes. That means we use color series: blue, green, yellow, orange, and brick red. Okay, blue, green, yellow, orange, and brick red. If we take a urine sample and if we add the sodium hydroxide and copper sulfate and we test as specified in the procedure. So if the urine is still blue, that means there's no glucose. And if the urine is somewhat green, so there can be traces of glucose. And the urine sample is will become orange. That means there's a moderate glucose concentration. Or if the urine sample will become a big red, that means there's a higher levels of glucose. So that is much severe. It means much severe types of diabetes can be in that individual. So likewise, we apply this test. Nowadays, it is not much used. Okay. Fructose solution green color because you know that reducing power of fructose is highest. Know that in fructose we have a primary hydroxyl group. So when there's primary hydroxyl group, that is easy to oxidize into carboxylic groups. Okay, then that is why fructose has a higher reducing power than any other sugars we tested. That is why it quickly become green color. Okay. So as I mentioned, මම දැන් කලින් කිව්වා වගේ මේක ටෙස්ට් එකක් විදිහට ගත්තොත් එම අපි Trommer's ටෙස්ට් එකක් විදිහට ග්ලූකෝස් කියන්නේ යුරින් වල ග්ලූකෝස් බලන්න ගත්තොත් එම අපි කලර්ස් හතරක් තමයි අපි මොකද්ද ඒ රේන්ජ් එකේ තියෙන එකක් blue color අනිත් එක green color orange color brick red color. blue color තියෙනවා නම් යුරින් වල ග්ලූකෝස් නැහැ. green color තියෙනවා නම් traces තියෙන්න පුළුවන්. ඒ කියන්නේ ඉක්මනට දෙනවා. traces තියෙන්න පුළුවන්. يعني පොඩ්ඩක්. orange color තියෙනවා නම් moderate ප්‍රමාණයක් තියෙන්න පුළුවන්. brick red color තියෙනවා නම් ගොඩක් urine වල glucose තියෙනවා. That is quite severe. Okay. And the other question is, what is change in color by fructose while adding copper sulfate at Trommer's test? Yes. The answer is similar. Fructose has a higher reducing power than any other sugars, even than glucose. Okay, you know that. If we compare the reducing power of monosaccharides and that disaccharides, reducing power of monosaccharide is higher than disaccharides. And if we compare the reducing power of monosaccharides, it means glucose, fructose, and galactose. The reducing power is highest in fructose, and secondly in glucose, and thirdly in galactose. Okay. Actually, there's no specific temperature to heat in Trommer's test. We just use the open flame. Okay, and you know that using open flame, we cannot exactly maintain a specific temperature. So that's it. Okay, then so I will move on to the next practical we have, that is to test identification test for polysaccharides. Okay, before that. So we basically did the earlier experiments to most monosaccharides and disaccharides. So then we will go for the other carbohydrate category, that is polysaccharides. Okay. So what do you know about polysaccharides? What do you know about polysaccharides? Okay, so polysaccharides. Then what can you say about the reducing ability, the reducing power of the, will it act as reducing agent, polysaccharides? So for an example, consider about starch. Will it act as a reducing agent? Polysaccharides are not reducing sugars or reducing, they have no reducing ability. Okay, they cannot act as reducing agents. So if we, even though if we use starch or something, some other polysaccharide in the previous experiments, they will give negative results. Okay, and even for Iodine test, it will not give big precipitation. Barfoed test similar, Trommer test also similar. Okay, then in the polysaccharides, actually the experiment number 7 is intended to test the solubility of polysaccharides. Okay, basically in this experiment we're going to use starch. Okay, before talking about starch, solubility of starch, so what can you say about overall, as overall, the solubility of polysaccharide? Is it, will they dissolve in water or not? What can you say? Polysaccharides. Some are soluble, some are insoluble. Yes. In general, we cannot say that all polysaccharides are not dissolved in water. Okay. Some polysaccharides are dissolved in water, some are not, some are partially dissolved. Okay. For an example, silane and cellulose. Okay, cellulose are polysaccharides, and they will not, cellulose will not dissolve in water. So do silane. Okay, and for example, gum arabic and pectin are polysaccharides. They are fully dissolved in water. Even in cold water, they will dissolve completely. Okay, then if we consider starch-like thing, it will partially dissolve in hot water. So the solubility of polysaccharides is different, depend on the type of polysaccharide. Okay, but you know that most monosaccharides as well as disaccharide dissolve in water. Okay, but the solubility of polysaccharides is quite different, and don't judge the behavior of polysaccharides only by starch. Okay, in this test we basically use starch, but don't generalize the behavior of polysaccharides only by looking at starch. Okay, some polysaccharides are fully dissolved in water, some are not, and some are partially dissolved in water. Okay, but in the case of starch, that will partially dissolve in hot water, and you know that sometimes you may not have experience. Then if you come to university, you will definitely, boys will have experience, and you have to prepare paper. So definitely you know that. Okay, so so what do you know about starch? We talk about starch. What is the empirical equation for starch? What is the monomer of starch?
Glucose.
Yes, glucose. Glucose are the monomers of starch. So glucose are linked through glycosidic bonds. And there are two major components of starch, or two major polysaccharides of starch. Yes, amylose and amylopectin. Yes, as you correctly answered, the two polysaccharides in starch are amylose and amylopectin. Okay, usually I'll just give an overall idea about starch, so that will be useful in these experiments. That's why I'm telling those things. You know that the behavior and the characteristics of amylose and amylopectin are quite different. Okay, usually in starch, um, uh, 20 to 30% are amylose and 60 to 70% are amylopectin. Okay, so what are the differences between amylose and amylopectin?
Straight line.
Yes, what has the straight line or linear structure?
Amylose.
Yes, amylose. Then one has answered that amylose has 1,4-glycosidic bond. Yes. Then what can you say about amylopectin?
1,6-glycosidic bond.
Yes, 1,6-glycosidic bonds as well as it has a branched structure. Okay. Yes. Amylopectin 1,4 and 1,6. Yes. And amylopectin has a well-branched structure. Okay. So what is called the resistant starch or modified starch? And sometimes nowadays we talk a lot about resistant starch, and there are lots of properties and good properties, health properties. What is meant by resistant starch? What is the importance of having resistant starch in our diet? So if you don't know, please search about that. Resistant starch can act as prebiotics. Okay, you know that probiotics are the microorganisms that are available in our gut, and prebiotics are the substances that are essential for the growth of probiotics. So prebiotic and modified starch are prepared by increasing the amylose content of starch. Okay, so I told you that in general starch, naturally starch contain 20 to 30% amylose and 60 to 70% amylopectin. So if we increase the level of amylose, the properties of starch will quite different. And amylose has more crystalline regions, and amylopectin has more amorphous regions. Okay, so I'll not talk deep about that. You will learn in your upcoming semesters about those things in food chemistry or any other courses. Then in biochemistry, I will not talk deeper about that. I just want to give an overall about starch. Okay, and you know that starch existing in the form of granules. Okay, starch granules. Okay, so I will talk about the experiment, and then we will talk where in the needy places. Oligosaccharides belong to polysaccharide. Technically, oligosaccharides are polysaccharides. Yes, technically we can say oligosaccharides means in oligosaccharides it contains more than 2 monomers. So technically they are polysaccharides. Okay, so actually we generally categorize carbohydrate into 3 categories: monosaccharides, disaccharides, and polysaccharides. Oh, okay, so oligosaccharides, I can remember the exact 3 to 10 or 11. When there's 3 to 10, 3 to 11 monomers, we sometimes categorize them as oligosaccharides. Okay, but then when there's more than 2 monomers, we can technically say that they are polysaccharides. But in polysaccharides, there can be monomers ranging from 3 to several thousands. Okay, so and I don't know whether you have heard about degree of polymerization. So that is one property responsible for solubility of polysaccharides. Degree of polymerization, DP. If you interested, please search about that. Degree of polymerization means that it's a measure of monomer units in a polysaccharide. Okay, then if a polysaccharide contains 20 monomers, the degree of polymerization is 20. So usually when DP is lower than 20, that polysaccharide dissolved in water well. Okay, when there's the degree of polymerization, it means the monomer units in a polysaccharide is less than 20, that polysaccharide will dissolve in water completely. Okay, so when I will move on to the experiment. Polysaccharides are polymers of monosaccharides joined by glycosidic bonds. Unlike the sugars, polysaccharides are not sweet. Okay. Then in this identification for polysaccharides, we will use two procedures. Okay. And in this experiment, in the first procedure, you have to take a little amount of starch into a test tube, and you have to add a cold distilled water and shake well. And you have to examine a drop of suspension under the microscope and filter the rest of it, and label the filtrate, means the resulting solution from the filtering, as X. And you have to evaporate drops of solution X on a watch glass and note that there are no residue or only traces of starch remaining in the watch glass, indicating that the starch is insoluble in cold water. Okay, if we consider about this procedure, as you can see, as you know, starch it is not soluble in cold water. Okay, PT වතුරේ දිය වෙන්නේ නැහැ. ඇල් වතුර දිය වෙන්නේ නැහැ නේද? ඒක හොඳටම දන්නවනේ. පිටි වලට ඇල් වතුර ටිකක් දාලා බලන්න දිය වෙන්නේ නැහැ. Okay, so when we in this experiment we basically test that. Okay, we add starch to cold water and we filter that. Then we label the filtrate as X, and we take a little amount of that filtrate and evaporate that on a water bath. Okay, then when there is no starch, then all the water will evaporate and there will be no starch residues. That is quite obvious. Okay, මේක මහලුකු දෙයක් නෙමෙයි. තේරුම් ගන්න අපි starch කියන එක වතුරේ දිය වෙන්නේ නැහැ. ඉතින් අපි මෙතන experiment එකේදී කරන්නේ starch ඇල් වතුර පොඩ්ඩක් ඇල් වතුරේ දාලා අපි ඒක filter කරනවා. filter කරාට පස්සේ අපි filtrate එක අර ගන්නවා watch glass එකකට අරගෙන ඉවරලා ඒක තියෙනවා water bath එකකට. ඒතර water bath එක තිබ්බට පස්සේ අපි ඔක්කොම operate කරනකොට පස්සේ අපිට පේනවා ඒ watch glass එකේ ඉතුරු වෙච්ච මුකුත් නැහැ. ඉතින් තියෙන විදිහකුත් නැහැනේ, මොකද starch වතුර දිය වෙන්නේ නැති හින්දා starch ඔක්කොම අර filter paper එකේ රැඳෙනවා. එතකොට ඇත්තටම filter වෙන්නේ වතුර විතරයි. Okay, that thing we testing the procedure 1. Okay, so that is the kind of solubility of starch, testing solubility of starch in cold water. Okay, then මේකේ බැරි වෙලාවත් මේක මේ starch වතුරේ දිය වුණා නම් මොකද වෙන්නේ? So what can you say about that? If we if in here if we use hot water, so that means partially starch can dissolve in hot water. So when we filter starch, the solution, then we may have residues at the watch glass upon evaporating. පැහැදිලිද? මෙතනදී hot water පාවිච්චි කරා නම් සමහර වෙලාවට starch residues ඉතුරු වෙන්න පුළුවන් watch glass එකේ. මොකද starch partially soluble in hot water. පැහැදිලිද? Okay, so in the second procedure, please understand this carefully, even though this is simple, but I feel that some students will not get this point totally. In here, procedure above A means procedure 1. This means taking the starch and adding cold water. Okay, in this procedure, we have to take as we done in the last procedure, we have to take a little bit of starch and add cold water, and we have to boil that. Okay, then you will observe that it become thick and translucent. Examine a drop under the microscope and compare with the above. It means the that we, after adding water to starch, we examine that under microscope. Then after boiling the content, we again take a little bit to the microscopic slide and observe. Okay. Pour a portion of the suspension into another test tube, half filled with water. And after that, we have to get half of that boiled thing into a hot water and filter the dilute liquid and label the filtrate as white. And after adding the half of the boiled substance into hot water, and then we filter that and we take it as white. Okay. Allow the rest of the original liquid to cool and note that it is gelatinizes. Okay. Then we will observe what is happening in the other half of the boiled starch. Evaporate a proportionate amount of Y on a watch glass and compare the residue with that of X, indicating that the starch is soluble in hot water. Okay, then if we do the same thing for Y, it means if we evaporate Y on Y taken on a watch glass, then we will observe starch residues at the end. Okay, so it indicates that starch is soluble, partially soluble in hot water. Okay, so I'll show you the practical recording. So after that, we can discuss more.
Today's practical is identification test for polysaccharide. For that, we are going to take a little amount of starch into a test tube. And then we are going to add cold water into it. And then we are going to shake it properly. Next, we are going to take a drop from this solution into a slide using a glass rod. We are taking a slide. And then I am going to place a drop from this solution into a glass slide. And we are going to observe it through the microscope. Then we are going to filter this starch solution into a conical flask. We are going to allow it to filter. Now it's being filtered, and we are going to label this filtrate as X. And then we are going to take a few drops from this filtrate, and we are placing it on a watch glass. On water bath for evaporation. Then we are going to place this watch glass on the water bath. Now we are going to observe the watch glass. Here you may see there are no residues left on the watch glass. Again, we are going to prepare a similar suspension as in the previous procedure. First, we are going to add a few amount of starch into boiling tube, and we are going to add cold water into it. We are going to shake. Now we are going to heat this suspension in the Bunsen. While heating, you may see the mixture become thick and then translucent. You may see the suspension has become thick. Now we are going to take few drops from this mixture and we are going to place it over the slide. And then we are going to observe it through the microscope. Then we are going to remove half of this solution into a boiling tube containing hot water. And I am going to keep few amount in this boiling tube. Now we are going to filter this dilute solution. the solution, and now we are going to take few amount from this solution into a watch glass, and we are going to evaporate it on a water bath. Now we are going to place this watch glass in the water bath. Now we are going to take the watch glass from the water bath, and you may observe there are some residues remaining in the watch glass.
Okay. So you have observed that what we are what we have to do in this identification test. Okay, in there we observe starch granules in under microscope, and as I mentioned earlier, starch exists in the form of granules. Okay, starch has two components, namely amylose and amylopectin, and this amylose and amylopectin are in the form of granules. Okay, so different, you know that potato starch, different type of starch is there depending on the source. Potato starch, wheat starch, rice, corn, many types, and the granular shapes and sizes are different from source to source. Okay, so using this microscopic view, we can identify the type of starch. So potato starch has this kind of shape, and rice starch has this kind of shape, and maize starch has this kind of shape. So this is highly used in food analysis to identify food adulterations. You know that sometimes then food adulterations is done by food processors. For an example, sometimes that means food manufacturers and distributors, they add some unwanted materials. For an example, for tamarind powder, they may add colored wheat flour. කහකුඩු වලට පාට කරපු කියන්නේ පාට කරපු පිටි දාන්න පුළුවන්. පිටි කියලා කියන්නේ නිකන් පාන් පිටි. ඊට පස්සේ for chili powder they may add colored wheat powder to chili powder. කියන්නේ මිරිස් කුඩු වලට පාට කරපු පාන් පිටි දාන්න පුළුවන්. ඒතකොට ඒ වගේ දේවල් then if we the best way to identify food adulteration in for those kind of things, to just a simple thing, we can just take a microscope slide and see what type of granules are there. So if there are different granules, it means the in chili starch, it cannot contain with starch granules. So likewise, we can identify food adulteration. So that is highly used in food analysis. Okay, I just want to tell you the applications of this test. Okay, and in the as the first microscopic observation, we're going to take we have to be in this experiment, we take a drop of cold starch cold water suspension. Okay, so there you will observe this kind of thing under microscope. Under the 10 into 10 into 1 view, you will observe this kind of thing. Okay, it means under low power you will observe starch granules as this, the undamaged granules. Okay, so this is a microscopic view. So you will observe this kind of undamaged granules in the first starch in cold water. Okay, so can anyone tell why starch is not dissolved in cold water but in hot water. What can be the reason for that? Why is that? ඇයි පිටි තනි starch cold water වල දිය වෙන්නේ නැතුව hot water වල දිය වෙන්නේ? ඇත්තටම වෙන්නේ may until the granular forms remain undamaged, water will not absorb to starch. May granular forms තියනකම් water may starch වලට water absorb කරගන්නේ. Or water cannot form any kind of bonds with starch. Okay, then you know that in polysaccharides it may contain higher levels of OH bonds, and as a result of that, intramolecular interactions are higher in starch. Okay, so that is why this polysaccharide exists in granular forms. Okay, when water, when we add higher temperatures or hot water, these granules can damage. Okay, then when this granular forms damage, it will release amylose and amylopectin to water, and after that they can form hydrogen bonds with water at in amorphous regions. Okay, so that's the basically why starch is not dissolved in cold water and in hot water. Okay, so in the second case we observe a microscopic starch in microscopic view of starch in hot water. There you will observe this kind of swelled starch granules or damaged starch granules. Okay, so here you can see microscopic views of it. Okay, so basically you will observe the starch granules are swelled. It means it absorbs water. It means water has made bonds with these starch molecules. Okay, and the you will observe the starch granules have been damaged. Okay, so that is why starch dissolved in hot water. So, okay, basically until these granules remain undamaged, starch water is not absorbed to starch. Okay, so then what will happen if we boil starch contained in cold water? In the second experiment, in the second procedure, we do that. What will happen then? In the test procedure, the demonstrator told you that you will observe thick and translucent appearance. Why is that? What is the process of happen in that thick and translucent appearance? What is the name we use to tell that? You will observe gelatinization. Okay, so starch gelatinization is a process of breaking down of the intermolecular bonds of starch in the presence of heat and water, resulting in swelling of starch granules by absorbing water in an irreversible manner. Okay, so that happened when we do heating. Okay, in the second experiment, that's why I told you that carefully observe that. Carefully observe. So it in there the starch gelatinization will occur. Okay, so in starch gelatinization, this kind of a series of changes and alterations to starch granules will be happened. Okay, so raw starch has this undamaged granular structures. While heating, this granular starch granular forms will swell by absorbing water and rupture them and Gelatinized. So gelatinization is a irreversible process. So we cannot revise that. Okay. So find about starch gelatinization, and in the test procedure also you have to observe what happened after cooling. Okay. So that thing is called retrogradation. Retrogradation. Okay. Retrogradation is the recurring and thickening of starch to give more crystalline regions upon cooling. So please, so this is, since this is not a theory class, I have no time to discuss with you about the theoretical things, but knowing about these things are very important. Gelatinization, retrogradation. Okay, find about these two terms, gelatinization and retrogradation, and why starch dissolved in water, hot water. So these are very basic things, but as agriculture students, this has many applications. As I told, just microscopic observations of starch granules is widely used to identify food adulterations. Okay, it is a very simple test to test food adulterations. Likewise, many applications are there. Therefore, find about those things. So don't just participate to these lectures, practical sessions. If you are enthusiastic and interested, please search about those things, because I have no time to discuss the theoretical things. Okay. So that's what we observe in the identification test for polysaccharides. Okay. The next experiment is iodine test. You have, I think you know about this test. This is also widely used to identify the presence of starch. Okay. So iodine forms colored complexes, blue, black starch iodine complexes with polysaccharides. Starch, most dextrins, amylodextrins, and glycogen will give positive results. Starch gives a blue color with iodine, while glycogen and partially hydrolyzed starch react to form red-brown colors. Okay, so iodine test is basically to identify the presence of starch. Okay, so in here we are going to use X and Y filtrate that we prepared in the previous practical. Okay, X means the cold water suspension, the filtrate of the cold water suspension, and Y is the filtrate of the hot water suspension. And a fresh dilute starch suspension into 3 test tubes, and we have to add 2 teardrops of iodine solution, iodine in potassium iodide. Dilute the inky liquid obtained in the case of Y until the blue color is clearly seen against the light. The blue substance is called starch iodide. Okay, boil the blue liquid, then cool under the tap water and make observations. So that is what we have to do in the iodine test. Okay, so if we talk about iodine solution, iodine is it does dissolve in water? Iodine, will it dissolve in water? Iodine iodine is not dissolved in water, and that is why in iodine solution it contains potassium iodide and I2, and the active form of iodine solution is I3 minus iodide. Okay, iodine I3 Ki and I2, it will form I3 minus. In iodine solution, you can see this active component I3 minus. Okay, so if I talk with you about the theory behind this test, iodine can form iodine starch complex with amylose. Okay, I told you that you know that amylose has a unbranched structure, as it is usually existing as a coiled structure. Okay, this iodine can go to this coiled structure, and that complex is dark blue in color. Okay, that is the principle of this test. Okay, you know that starch consisting of amylose and amylopectin, then iodide, I3 minus, and pentaiodide ions. I3 minus can react with iodine and can form pentaiodide ions also. These I3 minus as well as pentaiodide ions can form a complex with amylose. Okay, so that this is the reason for giving dark blue color for starch when we add iodine solution. Okay, so you will observe that when we as a part of this experiment, you have to boil the content in Bunsen burner. So when you are boiling, you will again observe that this will be colorless. Okay, you know that iodine is very volatile, and when we do heating, this iodide I3 minus will remove from these amylose structures, and upon cooling, the remaining iodide I3 minus can again go to these amylose structures, coiled amylose structures, and again form the color. Okay, so I'll show the video.
Today our practical is iodine test. For that we have iodine solution and 3 test tubes with starch suspension, and we have the test tube name as X, which we obtain the filtrate by dissolving in cold water. And then we have Y. It is the filtrate we obtain after dissolving in hot water. Now we are going to add 2 or 3 drops from iodine solution into these test tubes. You may observe the color changes. And then we are going to add iodine for the X. You may see a yellow color X. And then we are going to add few drops of iodine into Y. You may see it becomes to a dark blue color. You may observe that only X appears in yellow and the other 2 test tubes, fresh sample and the Y turn into dark blue. Then we are going to dilute this Y until we get the clear blue color solution. Now you may see the color is little bit done right now. And now I'm going to observe the color change. Now you may see the solution has become colorless, and now we are going to cool it under the tap water. You may see that the solution again becoming blue color. Now you may see the color has again turned into
Know about this test, iodine test, and this will give positive results. That means blue black කද අපි කියන්නේ කළු නිල් පාට complex එකක් දෙනවා starch තියෙනවා. Okay, so that is a identification qualitative test to identify starch. Okay, so I will not discussed far on qualitative analysis of carbohydrates. I'll stop from here in this session. We will meet in Friday, and if you have any questions, feel free to contact me at any time. Okay, then thank you for your patience and kind cooperation. Thank you.